Hello, good morning, all of you. We are studying history and milestones in microbiology, and in this particular topic, we are studying the contributions of different scientists, including Antony von Leeuwenhoek as well as Louis Pasteur, Fleming, Ivanovsky, etc. So, the contributions in the sense the inventions made by or discoveries made by different eminent persons for development of microbiological science. So, in previous lecture, we studied that Antony von Leeuwenhoek he was the cloth merchant actually, and but he discovered different lenses and he had a hobby to grind different lenses and he assembled the lenses or lens system and he observed the small animals or small organism and later on he described it as a bacteria. So in this way by discovery of this microscope at least we can study the small things in the sample. So this was proved by Antony von Leeuwenhoek and the different shapes of the bacteria, rod shape, cocci shape also he observed and he recorded his observation in his letters so that now we can easily that means we can easily find the information regarding the shapes of the bacteria by the observations made by Antony von Leeuwenhoek. Now the second eminent scientist we are studying here, we are going to discuss the contributions of famous eminent scientist that is Louis Pasteur. So if you know Louis Pasteur, he actually is known as father of microbiology because so many contributions, so many important contributions were made by Louis Pasteur. All of you know nowadays we are using pasteurized milk. The method of pasteurization was also discovered by Louis Pasteur for the preservation of wine and beer at that time. But nowadays we are applying this method of pasteurization for the preservation of milk in order to preserve the milk for in good condition. So we have to heat the milk sample in at specific temperature and we have to hold it for a specific time period like that. So this method was first time discovered by scientist Louis Pasteur. His time period was 1822 to 1895. This was the period of Louis Pasteur and during his lifetime. Due to constant and countless efforts or endless efforts and devotion as a dedication towards the subject, towards the microbiological science, he made so many discoveries, so many contributions for development of this science. So in 1856, he solved the problem of wine and beer industry. The beer and wine industry was facing a problem of sour wine. So we know that in France, the wine and beer industry was the major part of the country economy, and at that time what happened? So the souring of wine took place, and therefore the industrialist has set back because the wine and beer was not sold in the market, and therefore that means that was ruining the industry. And therefore the industrialists came to Louis Pasteur. Actually Louis Pasteur was chemist and he was asked to work on this particular problem, why the souring of wine take place and why the wine is spoiling. So the spoilage of wine and beer was studied deeply by Louis Pasteur and he discovered and he studied the major cause of spoilage of wine and beer. So firstly he studied the manufacturing process of wine and beer, and then he observed that the small organism, that is the microbes, they're responsible for fermentation of fruits and grains resulting in alcohol. So we know that wine and beer, these are the fermentable products, and by using grape juice or fruit juice and by fermentation method there is a production of these beverages, wine, beer, alcohol like that. So he studied the fermentation method in detail as well as he also studied the microorganism used in the fermentation process. And then he suggested the use of proper microbes for getting good product of wine as well as beer. So he discovered that in order to preserve the wine and beer in good condition, it should be pasteurized. That means it should be heated for some time and after then cooling and storing it proper place. And by using proper microorganism for the fermentation of this product, we can produce a very good quality of wine and beer. So this was proved by Louis Pasteur, and the same suggestion was applied. It was taken into consideration by the industrialists in order to prepare good wine and beer of good quality. So this was the first discovery made by Louis Pasteur. So this is a very great contribution made by Louis Pasteur. The secondly, he made certain conclusions regarding the fermentation. So we know fermentation is the process in which there is a raw materials are used. It is also a ATP generating process, no doubt. And here the production of certain alcohols, beverages is there in fermentation process. So he proved that each fermentation process requires specific condition, and alcohol producers grow well in acidic condition while lactic acid producing organism grow well in neutral medium. One thing we should remember that in order to get a particular fermentation product of good quality, it is necessary that the fermentation conditions should be favorable. They must be optimized, and then we can, after applying certain typical temperature, pH, as well as by using the raw material necessary for fermentation process, then we can use the method and then we will get a product of good quality. So it is necessary that certain fermentation conditions should be particularly, they should be provided there, and then only we will get the particular product. So in alcohol production, we have to use the organism which is growing in acidic condition, and for lactic acid fermentation, we are using the microbe which can grow in neutral medium. So it means that the pH of the medium should be also adjusted, it should be also proper, and it is depending upon the product which you are producing by fermentation process. So depending upon the type of the product, we have to adjust the pH of the medium. It may be acidic, it may be basic, or it may be neutral. Accordingly, we have to use again the microorganism for the fermentation process. So this thing was also studied by Louis Pasteur, that use of proper medium, proper microorganism, proper pH for the fermentation. He discovered existence of anaerobic bacteria. So what is meant by anaerobic bacteria? For the first time, Louis Pasteur reported that certain bacteria are anaerobic. Anaerobic means they are not having, they are not respiring in aerobic condition. They do not require oxygen for the respiration. So those bacteria which do not require oxygen for the respiration, they are considered as anaerobic bacteria. Aerobic and anaerobic. Aerobic means they require oxygen for the respiration. Anaerobic means they do not require oxygen for the respiration. So he discovered the existence of anaerobic bacteria also, and in 1864 he proved the theory of spontaneous generation by using swan neck flask. We will discuss later on what is spontaneous generation and how this theory was disproved by Louis Pasteur. So in order to disprove the theory of spontaneous generation, he used swan neck flask. स्पॉन्टेनियस जनरेशन थेरी म्हणजे नॉन लिव्हिंगपासून लिव्हिंग थिंग निर्माण होतात किंवा जन्माला येतात ही सांगणारी थेरी म्हणजे स्पॉन्टेनियस जनरेशन. पण ही जी थेरी आहे ही पाश्चरने डिस्प्रूव्ह केली, अमान्य केली, आणि ती काही एक्सपेरिमेंट करून त्यांनी केलेलं आहे. सो एक्सपेरिमेंटली ही प्रूव्ड दॅट द स्पॉन्टेनियस जनरेशन थेरी इज नॉट देअर. लिव्हिंग थिंग्स दे आर क्रिएटेड फ्रॉम लिव्हिंग थिंग्स ओनली. सजीव प्राण्यांपासूनच सजीव निर्माण होतात. सो निर्जीवांपासून सजीव निर्माण होत नाहीत ही थेरी त्यांनी डिस्प्रूव्ह केली. सो फॉर दिस पर्पज ही यूज द पर्टिक्युलर he conducted one particular experiment, and in his experiment he used swan neck flask. So the use of swan neck flask was first time used by Louis Pasteur, and he disproved the theory of spontaneous generation. In 1865, he proved germ theory of disease by showing role of protozoa in pebrine disease of silkworm. Silkworm, all of we know silkworm is mainly cultivated in order to get the pure silk, and silkworm industry is also there. So what happens, silkworms they get infected and the pebrine disease was there in the silkworm. So if the pebrine disease is there and if silkworms get affected due to this pebrine disease, there is no production of the silk, and again silk industry also get ruined. So this problem was again solved by Louis Pasteur. He studied the pebrine disease in detail, and then he suggested the farmers who are cultivated silkworm to utilize caterpillars in order to avoid the infection by the pebrine, the causative agent of pebrine, or in order to avoid the infection, in order to avoid pebrine disease of silkworm. So Louis Pasteur suggested use of caterpillars in order to avoid the pebrine disease. म्हणजे सिल्कवर्मला जो रोग होत होता पेब्राईन, त्या रोगापासून त्या सिल्कवर्मचं प्रोटेक्शन करण्यासाठी त्यांनी लुईस पाश्चरनी काही कॅटरपिलर्स वापरायला सांगितले फार्मर्सना, की ज्यामुळे तो डिसीज त्या सिल्कवर्मला होणार नाही. जर सिल्कवर्मला तो डिसीज झाला तर आपल्याला जे सिल्क हवं आहे त्याचं प्रोडक्शन त्या ठिकाणी योग्य प्रमाणामध्ये होणार नाही. म्हणून हा डिसीज थांबवला पाहिजे, या डिसीजपासून सिल्कवर्मचं प्रोटेक्शन व्हायला पाहिजे, यासाठी लुईस पाश्चरनी कॅटरपिलर्सचा वापर करावा असं सुचवलं. So that was the again germ theory which was put forth by again Louis Pasteur, and he stated that the germs are responsible for infection and for spreading the disease. So he suggested the use of caterpillars in order to avoid the pebrine disease. Now Pasteur also discovered the silkworm farmers could eliminate pebrine disease by using caterpillars for breeding stocks. जे ब्रीडिंग स्टॉक्स आहेत त्यामध्ये म्हणजे सिल्कवर्मचे, तर त्या ब्रीडिंग स्टॉकमध्ये जर कॅटरपिलर्सचा आपण वापर केला तर त्या ठिकाणी पेब्राइन डिसीज होणार नाही आणि चांगल्या प्रमाणामध्ये आपल्याला सिल्क किंवा रेशीम हे आपल्याला मिळू शकेल. दिस वॉज सजेस्टेड बाय लुईस पाश्चर टू द फार्मर्स, अँड ही ऑल्सो डिस्कवर्ड प्रिन्सिपल्स ऑफ ॲक्टिव्ह इम्युनायझेशन. So in 1880 he isolated the organism causing chicken cholera. He did again one experiment on chickens, and he discovered the principles of active immunization. It means that if the chicken cholera was studied by scientist Louis Pasteur, and he discovered that if it is the causative agent is already inoculated in the person, the immunity will be there in the person who have inoculated with the antigen or the causative agent already. So immunity or antibody production is there, and then the person will not be again get infected if it is already immunized. This is the principle of active immunization. So he did the experiment on chickens, and he studied chicken cholera, and then he observed that some set of the chickens who have already immunized with the causative agent, they get saved and protected, but those chickens who have not immunized, they got died. So in this way, by conducting the experiment, he proved that the active immunization is necessary in order to avoid the infection. So the principle of active immunization was also discovered by Louis Pasteur. Then the most important thing is that in 1881, Louis Pasteur prepared the vaccine to prevent anthrax disease in sheep. Now you can understand pebrine disease, anthrax disease, souring of wine, then silkworm disease. So many diseases he studied during his lifetime, and not only he studied, but he also suggested certain remedies as well as certain types of the treatment and the use, and again different types of the immunization you can say in order to get protection from this infection. So in 1881, he prepared the vaccine. Vaccine means attenuated or half-killed suspension of causative agent. That is the vaccine. Causative agent of what? Causative agent of disease. So we are preparing vaccine means we are actually isolating the organism causing disease. We are going to half kill it, and we are using either phenol formalin in order to prepare the suspension, and this type of the vaccine is inoculated. So vaccine is nothing but it is the attenuated or half killed suspension of bacteria or causative agent, you can say. So in 1881 he prepared the vaccine against anthrax disease. The anthrax disease was commonly occurred in sheep, and at that time what happened, if anthrax disease was there, different sheep get infected by Bacillus anthracis, which is the causative agent of anthrax. And therefore what happened, the sheep, that means their lives came into danger. The sheep were not saved. In order to protect the sheep from anthrax disease, Louis Pasteur discovered one particular vaccine in which he isolated anthrax bacilli, and he half killed that bacilli, half killed, not completely. So such type of the suspension was inoculated in sheep and some set of the ships, and then he proved that those ships who have already inoculated with the vaccine or attenuated suspension of bacilli, anthrax bacilli, they get saved and protected. So this was the principle of immunization. So in this way, the vaccine against anthrax was also discovered by Louis Pasteur in 1881. Then Pasteur prepared rabies vaccine also. See here, he studied pebrine disease, he discovered anthrax vaccine as well as now rabies vaccine. Rabies is a disease which is commonly occur in dog, and when the dog is infected by rabies or rhabdovirus, the dog runs amok in the street, and there is a constant drooping of the saliva from mouth from the dog. So when this such type of the furious dog bite the person suffer from hydrophobia. That means the rabies when occurs in case of animals, it is called as rabies. But whenever the person get affected, it is called as hydrophobia, because the symptom which is shown by the person, that is fear of water. Hydro means water, phobia means fear. So hydrophobia means he is having fear of water. He cannot swallow, he cannot drink water, even he is subjected to intolerable thirst. So that type of a symptom was seen in case of person who is suffering from rabies. So such type of the disease was there, and during his lifetime, the small boy Joseph Mister was severely bitten by the furious dog. And then what happened? This Louis Pasteur, he isolated the rhabdovirus, which is responsible for rabies, and he inoculated in the sheep brain. The virus was inoculated in the sheep brain, and he continuously harvested that virus, and so through so many several passages of this brain of the sheep, he then harvested that virus, and then he prepared the phenolized or formalized suspension of the virus. And such type of the crude vaccine, crude preparation, he inoculated in that small boy Joseph Mister. So at least he have given 13 inoculation of such type of the suspension to small boy Joseph Mister. And surprisingly, the boy was saved by this particular disease. The boy get protection from the 13 inoculation. So in this way, the rabies vaccine was discovered by Louis Pasteur. And later on, the boy, whenever he grew, the boy is saved for Pasteur Institute. So this is the history behind this rabies vaccine. So rabies vaccine, anthrax vaccine, pebrine disease, souring of wine, fermentation process, pasteurization, these are the important discoveries made by Louis Pasteur. These are the major contributions of Louis Pasteur. So we can say that indeed the Louis Pasteur He has made so many contribution discoveries for development of microbiological science. Now the next scientist is Robert Koch. So Robert Koch, he was the German scientist, German bacteriologist, and Koch is remembered for his isolation of bacteria that causes anthrax, tuberculosis, and for the rigid criteria which proved guidelines to identify the causative agent of an infectious disease. The major contribution of Robert Koch that he studied different diseases, means he studied anthrax, he studied cholera, he studied tuberculosis, and he also discovered the causative agent of anthrax, causative agent of tuberculosis, and causative agent of cholera. So he discovered Bacillus anthracis, then he discovered Mycobacterium tuberculosis, then he discovered Vibrio cholerae also, and these three causative agents were discovered by Robert Koch, and he, as he discovered, he made a very great contribution, so he won the Nobel Prize for his contributions. Certain criteria were put forth by Robert Koch regarding the disease. So he found that he discovered the causative agent of anthrax, tuberculosis, and cholera. As I said, Bacillus anthracis, tuberculosis, and this Vibrio cholerae. He invented strict plate technique and used gelatin as solidifying agent. Strict plate technique, which is very important type of the technique in order to isolate the organism. So in microbiology, whenever we want to study specific or one kind of the microorganism from a mixed population, the first step we have to take is the isolation step. It means that from mixed population, we have to separately grow the organism of our interest, and that is the isolation method. So for isolation method, we can use strict plate method, four quadrant method, back and forth method. So like that, so many sticking techniques are there. So for the first time, Robert Koch used strict plate method. So what is a strict plate method? We can take wire loop, we can take the nutrient agar medium plate, and aseptically we are taking the sample, and from one corner of the plate we can drag the lines, and such a four quadrants we can make. So in fourth corner the suspension will be thinner, so that in the fourth corner we will get the isolation or isolated colony. So the best separation will be obtained in fourth corner. Okay. So this is the strict plate technique. म्हणजे जो ऑर्गॅनिझम आपल्याला स्टडी करायचा आहे तो मिळवण्यासाठी मिक्स पॉप्युलेशनमधनं किंवा वेगवेगळ्या ऑर्गॅनिझमच्या मिक्सचरमधून तर आपण स्पेसिफिक मीडिया वापरून स्पेसिफिक कल्चर टेक्निक वापरून आपण तो ऑर्गॅनिझम आयसोलेट करू शकतो. त्याला म्हणायचं स्ट्रिक्ट प्लेट मेथड. तर स्ट्रिक्ट प्लेट मेथड पहिल्यांदा रॉबर्ट कोचनी डिस्कवर केली. सो ही डिस्कवर्ड स्ट्रिक्ट प्लेट टेक्निक अँड ही यूज agar or gelatin as a solidifying agent. आपण मीडियम करत असताना मीडियम म्हणजे ऑर्गॅनिझमचं खाद्य की ज्या मीडियमवरती आपण ऑर्गॅनिझम ग्रो करतो ते मीडियम सॉलिड व्हावं म्हणून किंवा सॉलिडिफाईंग त्याचं व्हावं म्हणून आपण जिलेटिन किंवा अगार ह्या पावडरचा वापर करतो. सो वी कॅन यूज जिलेटिन और अगार नाउअडेज for the solidification of media. But for the first time, Robert Koch, he used gelatin or agar in order to solidify the media. आपण जे प्लेटिंग करतो मीडियममध्ये प्लेटमध्ये मीडियम जे पोर करतो ते मीडियम लिक्विड असतं. तर ते लिक्विडच राहिलं तर आपल्याला ग्रोथ व्यवस्थित दिसणार नाही. त्यामुळे ते थोडं सॉलिडिफाय होणं आवश्यक असतं. सो हू इज अ सॉलिडिफाईंग एजंट? agar or gelatin. So gelatin will be the solidifying agent here, due to which the medium becomes solid, and on the solid media we can stick the suspension in order to get the best isolation of the colony. Koch also published 4 important criteria called as Koch postulates. कोचने काही postulates मांडलेली आहेत रिगार्डिंग द डिसीज. डिसीज का होतो, कुणामुळे होतो, किंवा त्याचे जे काही क्रायटेरिया आहेत, गुणधर्म आहेत, त्याचे काही वैशिष्ट्ये आहेत ती त्यांनी मांडलेली आहेत. सो कोच used in proving an organism is the cause of disease. Every disease is caused by the organism. It may be bacteria, it may be virus, it may be fungi, इट मे बी प्रोटोझोआ, एनिथिंग. कुठलाही डिसीज हा एकतर बॅक्टेरियामुळे होतो किंवा व्हायरसमुळे होतो किंवा फंगल असेल किंवा प्रोटोझोअल असेल. म्हणजे कुठला ना कुठला ऑर्गॅनिझम डिसीजसाठी म्हणजे पॉझिटिव्ह एजंट हा असतो किंवा कारणीभूत होतो. तो बॅक्टेरिया असेल, व्हायरस असेल, फंजाय असेल किंवा प्रोटोझोआ असेल. कुणी ना कुणी यापैकी रोगाला कारणीभूत हा होत असतो. सो देर इज सम बॅक्टेरिया द रिस्पॉन्सिबल. Bacteria may be responsible, virus may be responsible, fungi may be responsible for disease. So these are the agents which are causing disease. So the organism, what are the postulates made by Robert Koch? The organism must be found in lesion. Whenever the person is suffering from any disease, an infection, lesions are there. So we have to isolate the organism from the lesion exudates. So the organism must be isolated from the lesion. Secondly, the organism must be isolated in pure form. So we have to isolate the organism in pure form. त्यामध्ये बाकीचे कुठले ऑर्गॅनिझम येता कामा नाहीत. प्युअर फॉर्ममध्ये आपल्याला तो जो पॉझिटिव्ह एजंट आहे डिसीजचा तो आपल्याला आयसोलेट करता यायला पाहिजे. अँड वेन इनॉक्युलेटेड इन टू ससेप्टिबल ॲनिमल, प्युअर कल्चर कॅन रिप्रोड्यूस द डिसीज. Whenever we are inoculating the same organism in a susceptible animal, the pure culture can reproduce the disease. So this is one criteria, which is important criteria for the disease. So we are using this criteria nowadays also. Whenever the any animal or any person is suffering from a disease is caused by some bacteria or virus. So these bacteria or virus, they must be present in the lesions, and we are going to isolate the organism from the lesion in its pure form. So whenever the same organism is inoculated in the persons, the same type of disease should be produced there. This is what third criteria made by Robert Koch. The organism can be obtained again in the pure culture from the inoculated animal. Again, whenever the animal is suffering from the same disease, same lesions are there. So from this lesion exudate, again we are going to isolate the same type of the organism in pure form. So these are the criteria which put forth by Robert Koch. म्हणजे काय, एखाद्या ॲनिमलला डिसीज झाला तर त्या लिजनमधून आपल्याला तो ऑर्गॅनिझम आयसोलेट करता यायला पाहिजे. तो ऑर्गॅनिझम प्युअर फॉर्ममध्ये आपल्याला आयसोलेट करता आला पाहिजे. ज्यावेळेला तो ऑर्गॅनिझम पुन्हा आपण ॲनिमलमध्ये इनॉक्युलेट करू तेव्हा सेम प्रकारचे रोग, सेम प्रकारचं डिसीज आपल्याला त्या ठिकाणी दिसायला पाहिजे. आणि पुन्हा त्या डिसीजपासून जे लिजन्स आहेत, सो अगेन द सेम ऑर्गॅनिझम कॅन बी आयसोलेट फ्रॉम द लिजन अगेन. सो दिस दे शुड बी आयसोलेटेड अगेन in their pure form. So these are the same criteria which put forth by Robert Koch. So the first thing is that he used stick plate technique for the isolation. Secondly, he used gelatin or agar as solidifying agent in the medium. Then he also proved that the organisms are responsible for causing disease. They may be bacteria, they may be virus or fungal, any type of the small organisms that are responsible for the disease. So germ theory was put forth by Robert Koch. Secondly, the Koch postulates that is regarding certain diseases, he put forth certain criteria. That criteria we also discussed just now. Then the next contribution made by this Robert Koch is that the Koch adopted meat infusion and meat extract as the basic ingredient in the culture medium. Meat infusion, that is nothing but the meat extract. Whenever we prepare nutrient agar, that is the medium for growth of bacteria, we are using meat extract. What is this meat extract? The extract by which is obtained from this meat, and it is the best growth factor, or it is a good source of the nutrient, or you can say vitamins and growth factors. So if we are using meat extract, beef extract in the medium, there will be the good sources, best sources you can say, for the growth of bacteria. So such type of discovery made by Robert Koch. For the first time, he used meat extract in the medium, culture medium. Culture medium means the medium which we are using for cultivation of bacteria. That is the culture medium. So the meat extract or meat infusion is considered as the major ingredient or basic ingredient for the growth of bacteria because it is the source of so many growth factor and other nutrients types. So this is one thing here. He developed methods of studying bacteria with a microscope. So we know that basically the microbiology is the science which deals with the study of small organism and which we can't see with our naked eye. So for the first time he studied that, he proved that without microscope we can't study. So we need microscope to study bacteria. So he was the first scientist who described the bacteria by using microscope. He developed the method of studying bacteria with microscope. Then he used for the first time nutrient agar and nutrient broth for the cultivation of bacteria. Just now we discussed that the use of meat infusion or meat extract in the medium, culture medium. The culture medium is nothing but nutrient broth or nutrient medium. This culture medium is very necessary for the growth and cultivation of bacteria. So for the first time Robert Koch he used nutrient medium for the cultivation of bacteria. So these are the contributions of Robert Koch and Louis Pasteur. So we studied that Louis Pasteur he made several contributions for the development of microbiology science. He discovered pasteurization, he studied febrile disease, he studied fermentation process, and he discovered the method of pasteurization for preservation of wine and beer, as well as he discovered the anthrax vaccine, he discovered rabies vaccine, then febrile disease of silkworm also he studied very well, and he disproved the theory of spontaneous generation. So these are the several discoveries and important contributions made by Louis Pasteur. Then we discuss Robert Koch. He used nutrient agar, nutrient medium for the cultivation of different bacteria, as well as he used gelatin as a solidifying agent in medium, meat infusion in the medium also, and then he also discovered that germ theory also proved by Robert Koch, and for the first time he used microscope to study the bacteria. So he discovered anthrax bacilli, he discovered tuberculosis bacilli, he discovered cholera bacilli. So different organisms or causative agents were discovered by Robert Koch. So these are the contributions of Louis Pasteur and Robert Koch. Next lecture we will discuss another scientist and their contributions in detail. Thank you.